innovative forms of knowledge exchange and technology transfer, and has promoted a large number of initiatives that solve problems in the global south. Today we have 3 speakers. They're going to touch topics such as biopesticides in the SADC region, regulatory science. As we know, without regulation there is no product in the market, and the urgent need for making more food and new varieties with our population increasing in number. And then, without further ado, I'll call our first speaker, Dr. Vittorio Venturi, please.
Thank you. Thank you very much, Luis. Really a pleasure to be here. Thank you very much to Africa Bio for organizing this event. It's my first time, and it's been really very good and very nice to meet lots of colleagues and new friends. Can I have the first slide, please? So, as Luis mentioned, we have a big challenge ahead. It's not this presentation. So we have a big challenge ahead. The world population, the FAO, the Food and Agriculture Organization, an agency of the United Nations, has estimated that in 2050 we will be almost 10 billion people. And current food production cannot sustain that. So we will need to increase food production by 60%. Keeping in mind that we have to do this with less cultivable, with less soil, less land available for agriculture, and with all the unknowns of climate change. So we're losing land, it's becoming too salty, there is environments which are becoming too dry and too hot. So you can see the scenario that we have ahead of us. Here, this is actually the slide of the FAO, and the 9 billion is now almost 10 billion. So how are we going to do that? And so science has a big challenge ahead. You know, the COVID pandemic has shown how science can respond and succeed. And we will have to do the same with this big challenge ahead of us. So in ICGEB, the institute that I work for, the International Center for Genetic Engineering and Biotechnology, we have 3 components, 3 research components, one located here in South Africa in Cape Town, one in Trieste in Italy, and one in New Delhi in India. Plus we have 66 member countries. So it's a beautiful, unique organization for South-South cooperation, and unique organization. We have over 50 group leaders. We have about almost 700 scientists working. It's a really unique organization to start collaborating, and also with our laboratories from our 66 member countries, all located in the south of the world, to work on this big challenge. Okay. So just, you know, we only have a few minutes just to just highlight some of the things we're doing in ICGEB. We have scientists working in Cape Town and in India, in New Delhi, trying to understand how plants fight abiotic stresses, how tolerance to abiotic stresses, especially saline conditions and also heat, and at the same time biotic conditions like resistant to pathogens. And these studies, together with many other studies around the world, will lead to varieties which are more resistant to pests. We need that desperately. And at the same time, we're also looking at microbiology. We know from the studies on humans that we humans live together with so many microbes. In fact, we have 10 times more microbial cells than human cells living together with us, which do very important things, especially in our gut. It's like an external organ to our body, okay? So, and you know, it does a lot of important things like help digest our food, produce vitamins, induce our immune system, and provide a protective barrier against pathogens. Now, plants, we're now studying the plant microbiome. So the microbiome is a word which has been coined recently, which is the total microbial population of a niche. The plant microbiome studies are now catching up quickly. just like what happened in Newman a few years back, where now many scientists are studying the plant microbiome, and we're seeing exactly the same what happens with humans. And the soil closest to the plant root, which is called the rhizosphere, is 2 millimeters of soil around the plant root, is basically the gut of the plants, right? It's like the external organ of plants. And what it does, as I've written here in this slide, it helps the plant, is this a laser? It helps the plant to grow. It helps the plant get nutrients from the soil. There's a lot of nutrients that the plant just can't get access to. The roots are too big, or maybe the compound that they want, or the element they want is insoluble, and the microbes will help get nutrients. They will induce immunity. They will help the plant resist abiotic stresses like saline conditions, like temperature, and at the same time provide a protective barrier against the plant. So we're studying all this in ICGEB in the 3 components. With understanding the science. Is a fascinating science to study, but at the same time we can use this information to devise ways to help plants grow more sustainable. So what we're doing, we are isolating, we're studying these communities through next generation sequencing technologies and computational biology, and we make networks like this to understand the communities of certain or the most important crops. We work a lot with rice, and now we're working with sorghum to try and understand which are the communities, how they're working, and what they're doing. This external organ, how does it work, and which are the members? Okay, and this is what we're doing. And what is important that we need to do it as a network, as this session entails, collaborating. We need science. When I started science many years ago, each scientist was really working in their own bench, doing everything, and it was very much an isolated activity. Nowadays, collaboration is getting ever more important for many different reasons. To get results, to compare results, and also you need people of different expertise. There is a lot of new machines, new technology. So this is what we're doing. We're creating networks. As you can see here, I just show you, we're looking at the gut of plants of coffee. So the rhizosphere of coffee, and we looked at them with laboratories, all, as you see here, all around the world, because coffee is important in many different countries, and we're trying to develop a product, or trying to understand the science, but at the same time develop a biofertilizer which farmers that grow coffee can use in order to reduce the use of agrochemicals. So making science more sustainable. At the same time, we're doing similar with, apologies for the slide, which is a bit small, with sorghum. We started a network in Africa where we are actively looking for funding, but each of these labs is initiating some research activity with the funds they have. But we're talking to a couple of institutions and applying for grants, so we can create a network of sorghum growers, of sorghum scientists, in order to understand this microbiology, how we can use it, how we can use this information. So it's fascinating science, and at the same time with potential application. So to finish up, apart from the science, what we also ultimately want to do, and things can go quickly, that's also exciting. By understanding this microbiology, and also we are also isolating and working with the live strains, especially bacteria, we can improve soil health, and we can revitalize soils which maybe are tired and have lost biodiversity in their microbiomes. We can improve yields by supplementing these soils with the microbes which sorghum, for example, and coffee likes the most, and they have intimate relationship with. And we can improve plant and stress response and abiotic stress response. And we're collaborating with companies, and we intend to collaborate with companies in sorghum that will provide these, for example, these microbes that we can coat seeds. We can put them on soil directly, or we can even spray them in a foliar way. So I will end my talk here. So I hope you've got my message on this important topic, and how important it is to team up. And because analyzing lots of different soils, the same crop or the same plant grown in lots of different soils under lots of different stresses, we can get a lot of information on the microbiology, and then use that to devise products and ways to make agriculture more sustainable. Okay, thank you. We can, thanks. We have questions at the end,
right? Let's leave the questions for then we can interact more with the speakers. Then I'd like to call our second speaker, Dr. Dennis Ndolo, is the group leader of biopesticides at ICGEB Cape Town. The floor is yours.
I'll just grab my timer here. Thank you very much for attending this session. I was asked to be brief. I'll try to be as brief as possible, however long it takes. So I hope you'll bear with me, and I also hope I'll not succeed in putting you to sleep. Just trying to ensure my timer is on. Okay. So my presentation is going to look at how to enhance market access for African agricultural produce through harnessing the potential of biopesticides. We are aware that pesticides have made a significant contribution towards increasing or ensuring food security over the years. Synthetic chemical pesticides save us more than 30% of crop losses every year. However, over-reliance on synthetic chemical pesticides sometimes poses problems for trade. Now, just by way of introduction, before 2000, Sub-Saharan Africa was a net exporter of food. Now, the region, which is home to 13% of the world's population, has a net food trade deficit of 7 billion and actually accounts for less than 3% of global agricultural exports. In fact, Sub-Saharan Africa exports less food than Thailand, a country that accounts for just 1% of the world's population. There can be a variety of reasons why exports from Africa have been reducing, but one of them could be that some of the produce from African countries do not meet the residue standards in export markets. So you spray pesticides, the produce is harvested, but when it is sent to the export markets and tested, they find that the pesticide residues are above the limits that they would allow. So trying to export produce that exceeds residue limits can come with very significant consequences. Just as an example, this is just but an example, this is not something that has happened, but just as an example, citrus is a very important crop in South Africa. If they were to lose the market for citrus, they would lose over $54 million through trade. And if that happens, it also takes time to be able to resolve that residue violation. So it is very important that countries ensure that produce that they're sending to export market meet the requirements of those markets in terms of pesticide residue levels. Listed there are just a few pesticides that have been found in products from some countries in the region, not necessarily South Africa. And as I said, it is very important that countries ensure that they meet the residue requirements in the export markets. Different markets have different export requirements, and so the markets that a country is targeting, the country has to ensure that the produce they're sending to that market meets the residue requirements of those markets. How can we do this? One of the ways in which we can ensure that countries are able to better comply with the residue limits in export markets is through the use of biopesticides, the strategic use of biopesticides. Biopesticides, I'm sure most, if not all of us know, are pest control products that are made from living microorganisms, some minerals or other natural products. So that's what biopesticides are. And these products are very useful in terms of, if they're used strategically, they can really help countries ensure that they're able to meet the residue limits in export markets. We also call them germs for hire because they're essentially germs, but they help us to be able to control pests and hence get high yields but also meet pesticide residue limits. So I'm talking about strategic use. How can they be used? So there are various options. Growers can decide to use pesticides throughout, from the planting all the way to harvest. Now, one, that is not good agricultural practice, because when you use pesticides, you're supposed to stop at some point and give the pesticide time to degrade. That's the pre-harvest interval. But if farmers use pesticides, they get high yields, of course. The crops are not damaged, but then the chemical residue levels will be high, especially if they don't use the pesticides according to label instructions and they use them throughout the season. And so, even though they have high yields, they still would have difficulties accessing export markets. So that's one scenario. The other scenario is where growers use biopesticides throughout the season, so they don't use any pesticides. Now, what happens in that case is that you know that biopesticides are not, they don't kill as fast. Their efficacy is not as high as that of synthetic chemical pesticides. And so what then happens is that much as the crops produced solely using biopesticides will have low residue levels, but the damage will be quite high. So you'll have the holes on the fruit, or fruit damage. But even those produce, that sort of produce, cannot get to export markets. So even though you have low residues, the produce is such that you cannot sell it. So that's still a problem. The third scenario, and the one that we are advocating for, is where you use pesticides early in the season, for early season pests. Because most residue problems are caused by pests which come late in the season. So you use pesticides early in the season, but then late in the season, you then use biopesticides. Actually, what we are advocating for now is that when you're spraying pesticides, you can replace the last application with a biopesticide, just to ensure that you try and meet the residue requirements in export markets. So even though biopesticides are very important in this regard, they can make a significant difference, they occupy only 2.5% of the total pesticides market. This is due to a number of reasons, mainly to do with regulations. So many jurisdictions still don't have efficient regulatory systems for biopesticides. Actually, many jurisdictions still rely on regulatory regimes which were developed for chemical pesticides, which makes it expensive for product registrants, and it acts as a disincentive to the production of more of these products. There's also divergence in regulatory requirements between jurisdictions. So you may find that a country X needs data A, B, C, D. Country B needs X, Y, Z. So if I'm a product registrant, it makes it very difficult for me to access both markets because the data requirements are different. Another thing is that there are also, I hope the clock stopped. There's also lack of programs to try and integrate these products into good agricultural practices so that farmers get used to them. Farmers are generally risk averse. So relatively novel technologies, it takes a while for farmers to integrate them into their agricultural practices. So what we're trying to promote is to identify specific specific pesticides that can be used to address specific residue violations. And that takes me to a project that we are implementing, which is trying to address the problem of low export market access by some countries in Southern Africa owing to non-compliance with residue limits. And the project is looking at regulations, because I said regulations are one of the challenges. So we are trying to develop a system to harmonize regulations across the region so that product registrants find it easy to register these products in the various countries so that growers can be able to use them. So that's one component of the project. The other component of the project is we have actually identified specific residue problems, specific pesticides which are problematic for trade in various export markets. That table there, for instance, shows pesticides which are problematic for trade. And so what we are now doing is carrying out experiments. The last column shows the biopesticides. We're carrying out experiments to see if we can reduce the uses of those pesticides. We replace the last application with a biopesticide and see whether we can still get enough yield but comply with the residue limits. Countries should be able to do these pesticide studies themselves, to be able to develop these residue mitigation strategies themselves. And so one other thing that the project is doing is to provide capacity building for researchers in the various countries to be able to carry out these studies. That slide basically shows that all these things that we are talking about doing could ultimately result in more biopesticides on the market, enhance their use, because we also be providing training to farmers on how to integrate biopesticides into their normal crop production practices. We'll have less chemical residues, enhanced compliance with MRLs, and hence increased trade to various export markets. For this to happen, collaboration remains key. I know this session is about collaboration, and so we are working with various countries and various other regional and international organizations to meet the objectives of this project. Thankfully, I did not succeed in putting you to sleep. Thank you very much for listening to me.
Thank you very much, Dennis, for the nice talk. I'd like to now call Dr. Wendy Craig. Is the group leader of the ICGEB Regulatory Science in Trieste.
Good afternoon, everyone. I think I'm the shortest person here. So anyway, I also want to thank the organizers for this very generous invitation to allow us to present some of our work. I've done my very best to try and fit it into the title of the session, but if you'll forgive me if I wander a little bit astray from the directions that we were given for this session. But I want to also thank my good colleague and friend Dennis Ndolo for introducing the topic of biotechnology regulation, and which is the main one that I'm going to talk about, I hope, if we have the slides up soon. And why we want to focus on biotechnology regulation from our product development side is that in the absence of a regulatory framework, it's very difficult to, in the very least, think about having your product approved or enter that particular market, because regulations at their finest will be a way of assuring your customers that the entity has been approved because on the level more or less on safety more than anything, and it's compliant to various other rules that allow you to export or to enter other markets that you may not be able to do in the current regime. And hopefully, although I'm a bit staccato with this, hopefully this becomes clearer as I go through a number of these slides. But the key point is that in the absence of regulations, even if you have a really good idea, you can only go so far with the research. And even your research will fall under some other regulations as well. So everything that we do in a commercial sense will fall under regulations. And so therefore, Dennis has already pointed out, different locations, different jurisdictions, we call them, different countries have their more or less their own sets of regulations that were pertain to their certain conditions, what they want to achieve for their population with the applications and the products that they would like to have access to. And sometimes there's not a lot of crosstalk between what is required in order, in terms of data and safety levels, in order to get your product approved in one market as compared to another one. And so what we've been doing in ICGEB is to try and address that dichotomy and to help novice regulators, especially for those sectors in which maybe regulations didn't even exist because you're talking about maybe an emerging technology, or for which there's not a big head of steam in terms of expertise in the regulatory domain in order to allow those products to be authorised for entry into your markets. So I'm going to take a few minutes just to go over some of that here. In order to try and fit in with the other presentations in this session, we've been talking about agricultural products. So, of course, biotechnology can be applied across the life sciences, and you can have a whole range of products. But I'm going to focus for this afternoon's presentation on those that are more obviously in the agricultural sector. And I've chosen GM crops here. You're not really meant to take on board much of what I've presented here, but essentially, this is to show you that in the development of even a new variety of a crop in any sector of the world, is not something that you just do overnight. The whole product development stream is very extended. And I'll draw your attention to the green triangles towards the bottom, because for one particular variety to be approved, it can be anywhere between 10 to 20 years, depending on what we call the host platform, which species of crop that you're trying to modify and then get approved for entry onto the market. It averages around about 15 years, and over that 15 years, this is now old data, but a couple of years ago, it would take anywhere up to $140 million to get one GM variety approved. In this case, it was in the USA. And much of those development costs is not because of the protracted time, but it's more to do with the amount of data that's necessary in order to establish your safety levels and in order to gain authorization to go from the early phases of gene discovery through to implantation and insertion into your existing varieties, such that ultimately you grow them from the glasshouse into field trials, ultimately into multi-trial locations, such that you're generating all of this data to make the regulator confident that what you're going to hopefully have permission to put onto the market is absolutely safe in terms of not just consumption by your end customer, but also in terms of the interrelationships that it will have when it's being cultivated in the field. And throughout this whole process, there are different degrees and different types of regulations that each of the phases will fall under. And I've highlighted them here at the bottom of the slide that you probably can't see from the back of the room. Also, I found by speaking with my good friend Mr. Google on the internet, I found this cartoon of what the GMO application process is actually here in South Africa. And it's actually quite protracted. However, it's not unique. Most of the regulatory processes across the world are quite protracted because it goes through this phased approach. But this one in particular is for the end authorization to put it onto the market, the seeds in this case. And the data that has to be generated is submitted, it's checked over for veracity and whether you've ticked all the boxes of the type of data that's necessary to be looked at, that describes your product to the fullest extent, and then that's shown to various different experts that will be called upon depending upon the characteristic that you've put into that crop and those experts who are very familiar with that crop. And then they'll come up after another extended amount of time, they'll come up with their opinion on how well you've characterized your product such that they have faith in how you've analyzed those results, that you have demonstrated that the safety level has been achieved such that they can recommend to another body of experts that it should be authorized. And then once that body has looked at your recommendations, from that they can either agree with the technical body or they can disagree. And what tends to be is at the end of that process, it will either be approved or not. So this is actually the last 20th, or the 20% of the time of the development is actually trying to get this ultimate approval because of the amount of data that you need to produce. So, in real terms, what does this data look like? In different parts of the world, You see in the top left-hand box, that's the USA, and top right is the EU, bottom left is Japan, and bottom right is Korea. These are the only ones I could get photographs of the exact same variety that's going before these regulatory officials. And you can see that the data not only has to be presented slightly differently, but also the amount of data varies per country. And this is just for the very same variety, looking for approval in different parts of the world. And this is why it takes so long to generate the data, because they're going to ask quite searching and probing questions about it, and it takes a long time to review this data as well. So, if you can even see this, these are some of the golden principles when it comes to trying to regulate by technology. And it's for these very reasons that ICGEB, we're trying to instill these values into the various countries to which we serve and we give assistance to when they're developing their regulatory frames. And you can see yourself on the screen those elements. Another aspect of biotechnology regulation is such that if you are now, as a government, you're trying to put together a regulatory framework for a new emerging technology, you may not be the first country in which you have scientists and product developers that are looking at that new technology. It could be that through international collaboration among scientists, other regulatory bodies have also had a look at it before you. And so what's very useful and with the perspective of a product developer is crucial is that there's a lot of crosstalk between those regulators such that there's homogeneity between their data demands and how they perceive the characteristics of your product such that you're looking for consistent approaches to the regulation of your product. The worst thing that you want is another sense that Dennis mentioned, is that one country demands a whole host of data requirements, and then you go to your other potential market and they ask for a whole different set of data in order to assure safety there. So what we do as an organization is try to take out the core elements that are common to each, such that they have a bearing on international standards, and so that if we teach those international standards for those type of products, then it ensures that within a particular region, and we could talk about here the subcontinent of Africa, there's consistency across the regulatory approach such that hopefully the ultimate goal is that if a product is approved in one country, it will have an easier route to getting approval in a neighbouring country. So that's where we come into the whole picture. And we do this through what we call capacity enhancement activities. And this is where I hope it becomes clear why we're talking about South-South collaboration here. Because in the first instances, most of the advanced approaches towards the regulation of the technology were in those markets where they have far more products. And so there's more regulatory experience. They're dealing with far more applications per year than other parts of the world. And so this has tended to be in North America and in Europe and in Australia and New Zealand, that they've been the first ones to look at most of these emerging technologies. And it's also because most of the multinational corporations who are developing and applying these technologies, that's where they're based, and that's where their home markets have the greatest ability to recoup the costs of developing these products. And that's also a reason why many of the needs within the subcontinent of Africa, sub-Saharan Africa, have essentially been ignored by multinationals. There's a number of other reasons as well, because they have to recoup the development costs. They cannot charge a price in those markets in order to sufficiently quickly recoup their costs. And so that's another reason why there's less activity here in sub-Saharan Africa. And another one is just from my very first point, in the absence of these regulations, they don't want to expose themselves to potential liabilities if it means that in some of their new markets, they're asking for different safety levels. And what they had been assured was safe in one market may not be as safe in another market because of this inconsistency between approaches in regulations. So again, this is where the ICGEB, we've been working quite extensively these past 10, 15 years now. And we've been not only providing capacity enhancement activities to individuals who are beginning to become regulators, but also to government departments or government regulatory agencies whose mandate has expanded in order to cover these new emerging technologies. So we do it therefore at the individual level as well as at the institutional level. And what we're trying to do there is to assist them in being able to develop their own regulations. So we don't come in with a whole list of, a checklist of documentation and that and say, like, if you do it like this, then it'll be fit. There's no copying and pasting with us. We do it through a highly consultative process from the grassroots up, and we say, okay, what do you want your regulations to achieve in your market? Are you going to be open to innovation, or are you more precautionary and you want to take it in a more phased approach? Or are there certain technologies that you really have no interest in for whatever reason? And we assist them in developing those frameworks that attain their national objectives. Now, obviously, those that are a bit more precautionary, we can provide them with tools in which they can more quickly become familiar with the new technologies, such that that precautiousness that they might have had at the beginning, they become more familiar such that they quickly accelerate through to a level of familiarity such that they're more open to looking at new and emerging technologies. So on the African continent, we have been working quite closely with the African Union's NEPAD, and under their auspices, we've been helping them have access to the different ministries that will host these new regulatory bodies. And we've been facilitating a kind of cross-sharing and networking between regulators, for example, that came from Canada, Australia, etc., to help build up this, overcome this initial inertia such that they have a regulatory head of steam spread across Africa. But what quickly became obvious is that there were some countries at the forefront and some countries that were behind in Africa. So obviously South Africa has been one of the ones really pushing ahead in the regulation of the technologies, but also have been Ghana, Nigeria, Burkina Faso, Ethiopia, Uganda, Kenya. And those ones tend to be at the forefront of being able to quickly take on our training because it's more familiar to them. And essentially the work that we've done with them is more or less to put them into more direct contact with other regulators in other parts of the world in order to crosstalk and assure that how they use international standards is consistent. But in identifying them, we've also seen that they are far more open to helping their neighbouring countries come up to their level as well. So what we've been promoting through our activities is this more south-south development. So, for example, we've worked very closely with the Ugandan authorities in order for them, give them priority such that then they can reach out to their Kenyan and Tanzanian and Ethiopian counterparts and give them a leg up such that in the regions of Africa they're focusing on similar products, especially in agriculture, and they're learning from one another. And all we're doing is giving them a helping hand. So it's less input now from the more North American Europe, and it's very much a groundswell of activity on the African continent. So we do this, not just this establishing networks, because up until very recently there's been very nebulous networks for regulators to do this crosstalk. So actually Africa is at the forefront now of that because there's the very unique part of the world in which these regulators actively meet with one another and learn from one another. So we like to think that we had a part in that, but it's obviously up to the Africans, and they take the greatest pride in what they've been able to achieve. And this is just my group. We're a very small group, but we rely on our good friends in the regulatory agencies to do most of the work for us. So hopefully I've hit some of the remit for this session. Okay, so there you go.
We have time for questions. Any questions? Yes, Dinakar, please.
You alluded to the fact that different countries have different regulatory requirements. As a result, you need to make a different set of documents in each country. That by itself tells regulation is very ambiguous process, which means implementing regulatory rules, we are doing injustice to the researchers, because there is no exactness about what has to be regulated, which is to be regulated. For example, say in India, GM crops are totally banned, but in Bangladesh, all of them are being cultivated. And so Indian researchers who develop transgenics are at disadvantage. How does one deal with it?
Okay, for good or for bad, the regulations that you find in particular countries are a reflection of the concerns and the desires of the people, or perceived to be, because they're put in place by government entities, and hopefully most of these countries are democratic countries. So what you're saying is true, but it's not always consistently true, in that some countries under some governments can be quite precautious, and then they bring in a new government and they're more open to accessing the technology. But the reciprocal aspect can also happen with the change of government, and that's also another aspect of why getting authorization can be quite protracted in some countries. So it could be that Bangladesh has decided as a country through its elected governments that they are now more open to the technology, and it could be that they're more open because they have a greater or a pressing need for access to the products. You mentioned India, but I would say even further off the scale is Europe. Europe has looked at the technology in the agricultural sector and have said there's no such pressing need, and so they have the luxury of being quite anti the technology, because they're able to pay for imports at this point in time. So I don't also want to give you the impression that there's fluctuations or less veracity when it comes to the standards within each countries. There are certain key elements that are consistent, and that's where your internationals come about, and you have to attain those standards when you're looking at the regulation. However, there might be nuances because of the characteristics of the government or the crop itself that will be added on. That will be your greatest sector of variability when it comes to looking at the regulations as a whole. So I like the fact that Bangladesh is open. They recently, in the past couple of years, they authorized the BT brinjal. But our bets from looking at it outside, we always thought the Philippines would be the first to authorize them, but their application got stuck in the review process, and the Bangladeshis were able to do it far more quickly. That doesn't mean to say they're less safe, they just had different national priorities when it looked at the technology.
On which something like UN dwells common for all countries, because all humans are same.
Well, one of my slides did say that having effective regulation should be a human right in order to facilitate access to these new technologies, yes. But maybe my colleagues have something to input.
I have a question for the colleagues. Dennis, you indicated biopesticides are good, but they have also limitations. And so if you have to work out a process of use of chemical pesticide and biopesticide in a systematic manner for implementation, it probably is okay for rich farmers who have a lot of land, but to work it out in developing countries where small farmers cultivate land and most of the time they are illiterate, it is very, very difficult to find a complex mechanism of combining chemical and biopesticides. So what is your experience of implementation?
Yeah, thank you very much. And I think you rightly point out that biopesticides still face challenges, and one of those challenges actually is cost. And biopesticides are still costly also because the scale of production of many companies is still very low. Many farmers still don't use them. So even if there were small growers who wanted to use biopesticides, the costs are still rather high, and that is why at the moment biopesticides are mainly used in niche markets, as you say, the large growers. So one of the ways in which we're looking at it is that if the scale of production of biopesticides can be increased by addressing some of the challenges that make it difficult for these products to make it to market, for example, regulations. Companies find it very difficult to make these products because, one, regulations are a problem, and since regulations are a problem, there is not much of it that they can be able to make and scale up so that the products become a lot more affordable even for farmers. There's also an information gap because even many small farmers are still, recently we were in one of the countries in Southern Africa, and many of the farmers there are not aware of any biopesticide. So there's a problem of cost, but there's also a problem of information, and that is why one of the things that we are integrating in the projects that we are running is a training program. So we are actively involving the farmers, and one of the slides I mentioned that we are trying to develop a system where farmers are able to get to a point where they can incorporate this as part of their farming practices. So there's still a long way to go in terms of getting farmers to adopt this technology. And yeah, that would be my comment to that.
Can I add something to this? Also what I talked about, so having microbial products which can be used in order to have agriculture more sustainable and reduce, because even the products that I discussed about reduce the use of agrochemicals. To be realistic, they cannot replace fertilizers or biopesticides, or synthetic pesticides. They can reduce them. So we need an integrated. So I think it's very important for companies, and it's happening very much in the U.S. The companies that produce these microbial products, that they have assistance to the farmers. You don't sell the product or the seed and say and have a leaflet that you read. You really need follow up with the growers. Okay, I think the product is new, so the growers have to be made aware of these products and how they work, because sometimes they are temperamental. They're not always stable. So follow up of the product is with the farmer is very important in these microbial products. Yeah. You agree? Yeah.
I agree, and if you don't mind, I can just add one thing. So with regard to small growers, I think there are two categories in my experience. So you have situations where you have small growers who they grow their crops, but then they pull it together to sell to the companies that then eventually export. So for that category of small growers, it is a lot easier to get them to adopt biopesticides because it has a direct financial implication to them. What we have realized is that pesticide residue violations are more common amongst produce that is for the local market. So actually one of the things that we have begun thinking about is how can we look at residue levels within products within the local market and see how we can begin engaging with farmers to ensure that even products within which are not destined for export also meet those residue violations. And I think that is that still requires quite a bit of engagement.
Torio, I understood that plant microbiome is in the soil around the roots. So it is like a gut. Now, what are the molecules, presumably proteins, act as a receptor with respect to interaction between plant and microbiome for communication?
Well, that's a very good question. Firstly, just briefly, I've talked about the rhizosome, the soil closest to the root, so the gut. But there are also other microbiomes in the plant, like the on surface of leaves, or also the microbiome inside plants or in the seed as well. The parallelism I wanted to make is because the nearest soil to the root is really the gut, the most important one, the one that we can intervene most and have the most beneficial effects. I mean, what you've asked, Dinakar, is it's a very important issue. So what is the recruitment program of the plant? The plant, for example, exudes out of the root basically almost 15% of the carbon it fixes through photosynthesis, and also up to 20% of the nitrogen that it accumulates. And this also exudes a lot of amino acids, polysaccharides, and it does this to recruit and maintain the microbiome. Now, the signaling that takes place is a very important, we have beautiful examples from rather unique studies with rhizobia, with the root nodules and legumes, or with the mycorrhiza, and the communications being beautifully unraveled. And there are compounds made by the plant that are sensed by the mycorrhiza or the rhizobian, and there are compounds by the microbes that are sensed by the plants, and there's basically a beautiful, they're talking to each other, communicating to each other, and establishing their symbiotic relationship. With the microbiome, we are in deep sea. We are beginning to study this thanks to the new technologies that we have in our hands now, like genomics, transcriptomics, metabolomics, and fantastic new analytical chemical tools. We're now, a lot of scientists now beginning to look at what are the compounds that a plant produce, or the compounds that microbes produce, and how is this communication highways present in the gut of the plant. Because if my opinion is that ultimately what you really want to do, in my view, is not to add microbes, because the microbiomes are so complex. It's thousands and thousands of species that live together there. It's like a multicellular organism. And to think that to put one or two and you solve the problem is a bit naive, right? So really the long-term, I think, the long-term application is to instruct the plant, to modify the plant, thereby allowing the plant to signal to its soil to recruit the microbiome, because soil today is the most biodiverse niche we have on the planet. Now the soil has the most diverse microbiology, incredible. So the tank of microbes that the plant can have access to is amazing. So the signaling is I think the ultimate thing we need to understand. And I'm optimistic, and then if we understand the signaling, then we can make sure that the crops that we plant have a good signaling system in process to recruit a good microbiome and a healthy microbiome. So then the plant is stronger, healthier, can get more nutrients, can fight disease better. It's a bit like humans, if you have a good microbiome, if you eat healthily and if you do exercise, you're less likely to get ill. Same with the plant. We have to make sure that the plant is able to recruit a good microbiome so then it can grow well and can keep the pathogens away. And we humans need to understand the microbiome biology and the signaling and intervene on this. It's going to be a beautiful work to be done in the future, and I'm really optimistic because of the technologies and as what we discussed today, and the need for collaboration, because Dinakar, we need chemists, we need analytical chemists, we need molecular biologists, we need plant biologists. So we need to collaborate and create. On the technologies I work, well, I'm
Omics.
Yeah, I'm a geneticist and molecular biologist, so I'm old, so I started, when I started doing research, I sequenced all my genes by myself, without primers. I tell that to my students when they send their piece of DNA to sequence, and after 24 hours they are during their long coffee break say, where is my sequence coming back? I said, hey, wait a minute, I did everything by hand. So I was born as a geneticist and molecular biologist, so I studied bacteria, but now I'm becoming more of I'm using the genomics in my work, which is you know, we all know that a revolution has been that DNA sequencing today is so fast and so cheap. So we can do amazing things. We can make photographs, lots of photographs of all the microbiology, you know, can do functional genomics as well. So it's exciting.
Thank you.
Thank you.
And just a complementary question for you. Yeah, with all these omics in this microbiome field, how easy is to get funding for that? The companies are working on this as well.
Thank you, Luis, for this very painful question. I work in Europe. I was reading an article by 3 very famous scientists in the US. It was quite an old article now, maybe 70 years old. And they published how, for example, in the US, biomedical sciences is funded compared to plant sciences. Biomedical science is funded very, very well, and the number of PhD students in the US is increased almost, you know, incredibly in the last years. And it gets to the point now that maybe not all PhDs in the US can get jobs quickly in the biomedical field. In the plant science sector, it's been flat in the US. And, you know, we're talking about the US, the paradise of funding for research probably in the world. In Europe, you know, we have a lot of problems, as also my friend and colleague Wendy said. You know, we're very cold towards plant research. We don't even, you know, so it's very difficult. And it's an issue that it's something that concerns me. I think this was slowing down a lot. The urgent need, as I said in my slide, 60% more food for a growing population within 2050. It's a big challenge. And if we don't get the funding to do it, we won't get there. So the question you made, Luis, is I have a lot of problems in my country, in Europe, to get funding. I work a lot with companies, and you know, companies, it's obviously a way of funding, but they're very demanding. They don't give you a lot of money. They're very applied research. We need also fundamental research. So it's something that we need to lobby, and we need to, you know, the COVID has been fantastic to show us how the scientific world can mobilize, and with an extremely short time. I mean, it was a beautiful success story. We had, I don't know how many vaccines within 12, 15 months, how many antiviral drugs. So it's been a beautiful way the world has seen how science can respond to an emergency. We need to do the same pretty soon. We need to get moving with the agriculture. We're getting more food, more sustainable, in a much more sustainable way. We're going to get less land, climate change, more people. So we need to mobilize also for agriculture. And I hope we can lobby. And also the scientists studying the biomedical field, they need to let the governors and let the governments know that we need to fund much more agriculture and plant-related sciences.
Yeah, I'll just
You
add.
agree? Yeah, I mean, yes.
No, it's related to an earlier question. As I was sitting here waiting for the next question, I felt that I did not do sufficient justice to the question that Dinek had asked earlier about the challenges of biopesticides, especially for small growers. I just wanted to add that, I mean, farmers are generally risk-averse, you know, so they would like to stay with technologies that they are accustomed to. So small growers know that when they apply synthetic chemical pesticides, they'll get a good crop and high yields. So one of the things that we have also actively started doing now is to try and reach out to farmer groups in the various countries. So we have started with about 6 countries in Southern Africa to actively engage farmer groups so that we begin discussions on how biopesticides, biopesticides could contribute to ensuring that they produce crops which are healthy both for the local market, but which could also enable them begin to tap into export markets, because you have small growers who don't sell to export markets, but if they could use some of these products, then they can then begin forming networks which can enable them begin getting into networks that can enable them be able to export their produce. So the groups that we are engaging with now actually are farmer groups that bring together various farmers to try and begin this discussion so that they eventually appreciate how significant biopesticides can make in their farming operations. I think now that is enough justice. Yes, thank you.
I think we are running out of time. I once more like to thank our three speakers for the nice talks today and for everybody for the discussions. And with this, we just end our session. Thank you.